All right, welcome back. Welcome back. Our last session, I'm Chris Forsmark. I'll be one of the hosts. And
I'm Vijay Singh from Mayo Clinic, Arizona. Chris and I will be moderating this session. We would request that each of us be seated. Thank you.
all righty, everybody, thanks for hanging on to what will be a really interesting session. So this session is titled Exocrine Involvement in Type 1 and Type 2 Diabetes. And I'm going to introduce the first speaker, Brittany Brueggerman, who is from my home institution, the University of Florida. Go Gators. And she's going to talk about, as I said, her title is, at least the title is not terribly focused, and that's what is the role of the exocrine pancreas in type 1 diabetes. But we're going to learn a lot, I think, from Brittany, so I'll hand it over to you.
Okay, thank you so much. I'm really excited to be here, and it's been so much fun to have a whole day dedicated to stuff that I think about all the time, but everyone around me is like, oh, that's very interesting that you're looking at that. So yeah, this has been a lot of fun. But yes, my talk today, I tried to cover a lot of material because it is a very broad subject, but we will do our best. I'm looking at the role of the exocrine pancreas in type 1 diabetes, and thankfully for me, Dr. Wright set this up really well, so hopefully you've retained a little bit of the background information from his talk too. I don't have any conflicts, and I am on X and LinkedIn, and we should connect because, again, this is my fun little research area that all of you are also interested in. So definitely connect. There we go. Okay, so we already covered this, but I'm going to go over it again just to make sure we're all on the same page. So type 1 diabetes is an autoimmune condition driven by autoreactive T cells to the beta cells. It starts way before clinical diagnosis. So that's something that I think a lot of people don't realize is the autoimmune process typically starts years prior to the onset of clinical disease. And so you have genetic risk, which is typically driven by HLA along with some other genes. And then you have some sort of environmental exposure, we think, possibly a virus, some other things that we're all looking at, which starts the autoimmune process. Now, we know that is happening because we measure something called islet autoantibodies in serum. We typically measure around 4 of these. And if you have 2 or more islet autoantibodies that are positive, then you are labeled stage 1 type 1 diabetes. And we did that intentionally because you have about an 80% 10-year risk of developing clinical type 1 diabetes if you have 2 or more islet autoantibodies. So we think you will go on to develop clinical disease eventually if you have these antibodies. Then stage 2 type 1 is what I like to think of as the pre-diabetes of type 1 diabetes. That's when you have dysglycemia and islet autoantibodies. You don't meet criteria for diabetes yet, but you do have abnormal blood sugars. And then stage 3 type 1 diabetes is what we think of as classical clinical type 1 diabetes. So here are the kind of milestones that in my field everyone talks about in type 1 diabetes. So in 1921, we discovered insulin. In 1974, we discovered that autoimmunity was the driver of type 1 diabetes. In 1986, we had our first type 1 immune therapy trial. And then finally in 2022, very exciting, we have our first disease-modifying therapy in Tzield, which is a monoclonal antibody directed towards T cells. However, what we don't often talk about is even before the discovery of insulin, way back in 1909, we knew that people with type 1 diabetes had decreased pancreas size and acinar inflammation. So we knew this way back when. Then in 1925, we demonstrated that there is exocrine dysfunction in some type 1 diabetes patients. There was not a whole lot of interest in looking in this area until around 2012, which is when Dr. Campbell Thompson in my group found in organ donors with diabetes. Oh no, that didn't. Okay, there we go. In organ donors with diabetes. Sorry, it switched on my other slide. There we go. Okay. The pancreas weighs in organ donors with autoantibody positivity but no diabetes, so these sort of what we would call stage 1, they have a pancreas size that's 25% smaller. And so that's when everyone was like, oh, wow, this is actually happening early on in the process of developing type 1. And so we really wanted to figure out what was happening. So this is when we had sort of a renewed interest in this topic. Dr. Wright covered sort of potential mechanisms, but the case for potential developmental defects happening as a cause of exocrine dysfunction, again, we've learned today that exocrine and endocrine cells come from a common progenitor. There has been congenital exocrine hypoplasia reported in like a case report with someone with type 1, but it is not a common finding to have this. But there are abnormalities in first-degree relatives who do not have islet autoantibodies. And so that is a case for potentially there's something happening developmentally to cause this exocrine dysfunction. I guess, we'll talk about what we all think is going on, but I think there are probably multiple things going on, and I think a large part of it is in the insulin trophic effect, like Dr. Wright covered, but I do think other things could be going on as well. But another thing that we talk about is exocrine-endocrine crosstalk deficiencies. We cover this data a little bit, but in type 2 diabetes, there is milder but significant reductions in exocrine size and function. And I don't think we covered this, but some of that loss can be reversed with disease remission. And that's not necessarily exogenous insulin delivery. It's more, like GLP-1s and other medications and lifestyle changes that actually reduce or contribute to endogenous insulin secretion. So that's a case for potentially, again, the role of endocrine-exocrine crosstalk. There is greater atrophy in the pancreas body and tail, which has a greater proportion of beta cells. So again, a little bit of a case for that insulin trophic effect. Decreased alpha cell mass actually correlates with reductions in pancreas weight too, which is something that's a bit understudied at this point. However, a little bit going against this is that acinar cell number, which is actually Dr. Wright's data, is reduced rather than the acinar cell size in type 1. And so that goes a bit against the insulin trophic hypothesis, because you would expect maybe the cell size to be more reduced rather than cell number. And then there are, again, abnormalities in first-degree relatives and autoantibody-positive subjects who have normal blood sugars. Now, that's not to say that all of their insulin secretion is normal, but at least their blood sugars are normal, and a number of these don't actually even have autoimmunity yet. Exocrine inflammation or autoimmunity is another hypothesized contributor to exocrine dysfunction in type 1. So this is very interesting. When you look at a compilation of studies, there are exocrine autoantibodies present in about 20 to 70% of people with type 1 diabetes. And there's this whole list of them up here that you can see. A lot of these are not commercially available, so it makes it difficult to do further studies with this unless you develop kind of your own ELISA and do all this stuff. But it is really interesting that these different studies have found this. And then a couple of the studies have actually looked at it in relation to exocrine function, and it does seem like exocrine function is reduced in some of these studies where there are exocrine autoantibodies present. And again, these antibodies are actually also more commonly present in first-degree relatives of people with type 1 diabetes, so not only those who have clinical disease. Just to go over the data real quick about stage 0. So this is these at-risk people. So usually in these studies, they're either a high HLA risk or have a first-degree relative with type 1 diabetes that don't actually have islet autoantibodies present yet. And so a number of studies have shown a difference in pancreas volume and in function in these stage 0 at-risk people. And so again, data from the UF group, Martha Campbell-Thompson, showed relative pancreas volume is reduced in T1D first-degree relatives. PENO is the NDIA birth cohort study based in Australia, which is really interesting. And they showed that fecal elastase decline actually precedes the onset of islet antibodies in some progressors. So they don't yet have islet antibodies, but they have a lower fecal elastase value. Another study looked in the TrialNet cohort, which is this large type 1 diabetes clinical trial network that follows people over time who are at risk for type 1. And it showed that there are reduced amylase and lipase levels in type 1 diabetes first-degree relatives, but they only found this in adults and not children. And that's kind of where we're at with this. Some studies find that there is a difference in these first-degree relatives without antibodies. Other studies don't find a difference, and there's quite a bit of difference in the cohorts who are being studied. And one thing to know about type 1 diabetes is it seems like things are quite different in terms of autoimmune progression and sort of the phenotype dependent on age. And so age is kind of an important cofactor in a lot of these studies. The younger people tend to develop type 1 much faster and have a more acute progression of disease than adults who are diagnosed. In stage 1, there's a lot more consensus that we do have exocrine deficiencies and abnormalities when you have, and that's again when we have islet autoantibodies but no dysglycemia yet. So there are multiple studies that have shown imaging differences that we previously talked about with reduced pancreas volume. Dr. Wright also talked about how this does predict progression to stage 3, which is really exciting. And then other markers, so lipase and trypsinogen. I just found out that you guys don't really measure trypsin or trypsinogen very often, but we have found in type 1 diabetes research that trypsinogen and lipase tend to be lower in people with type 1 diabetes, and then even in these islet autoantibody normoglycemic individuals, they are lower, and they also correlate with stage of diabetes. So you can differentiate like stage 1 type 1 diabetes from stage 3 using lipase and trypsinogen. Our unpublished data, I don't know if I can use this. How do I do the clicker? Oh, this one. Maybe, here we go, okay. So this was a study that we did where we looked at young organ donors with type 1 diabetes and then young organ donors who were islet autoantibody positive but without dysglycemia. So we only looked at organ donors under the age of 30 just because older individuals tend to have a lot of pancreas pathology and we didn't want to kind of muddy the waters with that. And so under the age of 30, acinar atrophy and chronic pancreatitis, interestingly, was present in organ donors who had multiple islet autoantibodies, and even more so in young donors with type 1 diabetes. So this is the orange bars are acinar atrophy. This is type 1. This is multiple antibodies. And then the, white bars are chronic pancreatitis. There was, maybe even a bit more chronic pancreatitis in these multiple antibody positive individuals, but it was a small cohort, so you can't say that for sure. As Dr. Wright kind of described before, it's been really difficult to parse out stage 1 and stage 2. So is this exocrine atrophy and these changes, are they more associated with the development of islet autoimmunity or with loss of insulin as a trophic factor? And that's difficult to parse out with existing data, but we'll get a little bit, that's a hint to more to come. And then in new onset stage 3 type 1 diabetes, so clinical disease, these differences are even more pronounced. And there's actually a 30 to 50% reduction in pancreas volume and in pancreas function at the onset of type 1 diabetes. The pancreas has a large reserve, and so clinical function is usually preserved. So you're not going to necessarily see fecal elastase levels less than 200, but you do see a reduction in fecal elastase and in amylase and amylase is so-so, but trypsin and lipase, you see quite a bit of a reduction in those. Studies vary widely on whether or not clinical function is affected in type 1 diabetes. So, for example, should we be adding screening for exocrine pancreatic insufficiency in type 1 diabetes? It's hard to say because right now, there have been systematic reviews, and one systematic review showed that as many as a third could have exocrine insufficiency, but other studies show a much smaller percentage. And so I think we're still kind of trying to answer this question of, do we need to be screening people with type 1 for this? And if so, are there certain people who warrant screening versus others? I would say right now, if somebody has symptoms of exocrine insufficiency, it should be a consideration for people with type 1 diabetes, whereas a lot of clinicians will just think of celiac disease if there's any sort of GI symptoms. But if people with type 1 have GI symptoms, it's a consideration to screen for this. Then an established type 1, again, this is more data from Dr. Wright's group, so he's contributed quite a bit to my talk too. But there is a continued decline in pancreas volume even after the onset of type 1 diabetes. So especially within the first year after diagnosis, it continues to decline. And then in terms of duration of diabetes versus function, the studies are a bit mixed, but several studies do show worse function in adult patients who have long-standing disease. And then again, we talked a little bit about this, but in type 1 diabetes, organ donors versus controls, there are a lot of different changes in the pancreas. In type 1 diabetes, there were more acute and chronic pancreatitis, acinar atrophy, acinar fibrosis. We found periductal fibrosis. One thing that is interesting is that people with type 1 diabetes do not tend to have fatty infiltration of the pancreas, and so that is a change that we do not see in type 1 diabetes versus type 2 diabetes. So this is a study that our group did to try to better parse out this question of autoimmunity versus dysglycemia, and when are these changes happening in the development of type 1 diabetes. So we were looking at the T2D cohort, which is a very large birth cohort of children who are at risk for type 1 diabetes. And they are followed from basically birth. So the first samples are done at 3 months of age, and then followed out until at least age 12 or so. And so we wanted to try to figure out this question about autoimmunity versus insulin trophic effects, and then also just look at fecal elastase as a potential biomarker of progression to stage 3 type 1 diabetes. And you guys are the first ones to hear about the results, so, congrats. So this was a nested case control of fecal elastase through the pre-type 1 period in T2D. We basically designed this to be 2 different cohorts to try to answer this question better. So cohort A were very early progressors to stage 3 type 1 diabetes. So cohort A cases developed type 1 diabetes, like stage 3, like clinical diabetes, by age 4. So very early onset type 1. And then cohort B developed stage 1 type 1 diabetes, so islet autoantibodies, but they did not develop, so they developed stage 1 by age 4, but they did not develop any dysglycemia out to age 6 at least. And most of them developed dysglycemia. If they did, they developed it much later, so closer to that like age 10 to 12-year-old time period. And so we were going to compare fecal elastase in these 2 cohorts, and we hypothesized that fecal elastase would be reduced even before stage 1 in cohort A, and that the rate of decline in fecal elastase could predict stage 3, and that fecal elastase would be less impacted in cohort B, who doesn't have dysglycemia. So I think our findings are really interesting, but again, I'm in this space 24/7, but we did find that even at the 3 months of age time point, prior to the onset of islet autoimmunity, we did see differences in fecal elastase in cohort A at that time point. But as y'all will notice, these are not in the deficient range. And in our entire cohort, I think we had maybe 2.5% total of individuals who actually had a fecal elastase below 200. And so it was very rare to actually be deficient, but you did see lower values in these early progressors to type 1 diabetes. And so we saw a baseline difference, and then we also saw a more pronounced difference as time went on. So at the onset of stage 1, the difference was more pronounced, and then the onset of stage 3, it was actually the fecal elastase values were about half of that of the control individuals. And then in cohort B, kind of as we thought, there was no differences in the overall cross-sectional fecal elastase values between cases and controls. Next, we looked at the trajectory of fecal elastase over time. And so even prior to the onset of stage 1, so prior to the onset of islet autoimmunity, there was a downward trajectory in stage 1 in the cohort A individuals. And so they had this negative trajectory compared to cases. And then there was also a negative trajectory before the onset of stage 3. In cohort B, so we did not see a difference in cross-sectional values, but we did see a negative trajectory prior to the onset of stage 1 in fecal elastase values in cases versus controls. So there was a bit of a change, a negative change in fecal elastase prior to the onset of stage 1. However, when you compared trajectories, the cohort B decline was slower than the decline we saw in cohort A. This was not, I was hoping this would be better, but fecal elastase was only a modest predictor of the onset of stage 3 type 1 diabetes. And so this is something that now we will be looking at whether we can combine fecal elastase in combination with other biomarkers or markers of insulin function or clinical characteristics to see whether it improves existing risk scores for type 1 diabetes. But we wouldn't use it alone as a biomarker for prediction. I'm not going to cover this in detail, but we've also interestingly seen that trends in amylase and lipase and, trends in amylase and lipase are different in responders versus non-responders to immunotherapy in type 1 diabetes. And so this was looking at ATG and GCSF treatment. And we found that responders to treatment had preserved lipase and trypsin values, while non-responders and placebo subjects had declining lipase and trypsin values after treatment. And we saw actually a trend towards a baseline difference in lipase and trypsin values, but these were only I think the N was 35 in this study, and so we would need to do a lot bigger of a study to validate our findings there or see if there is actually a baseline difference. Okay, I am done. So to conclude, we have exocrine volume, histopathology, and function are all impacted in the pre-type 1 period. This is more prominent, as we've seen in other studies, in the later stages of type 1 and associated with glycemic changes, but there are baseline differences present in early progressors to type 1 and a downward trajectory in islet autoantibody positive non-progressors. There are potentially changes in exocrine functional markers in up to a third of type 1 patients, so something to consider, and they may be useful biomarkers of progression or response to therapy. And overall, we need a lot more work, so anyone who wants to join us is welcome. Thank you so much.
Thank you so much, Dr. Bergman. Our next speaker is Dr. Anna Kasu. She's Associate Investigator at the Translational Research Institute at Advent Health, and she's the Von Weller Family Chair for Type 1 Diabetes Research. Dr. Kasu.
Thank you to the organizer. Oh. Where's this? All right. Thank you to the organizers to inviting me to speak today and for challenging me to put everything in 15 minutes. So we are talking about adipose tissue now, and we heard a little bit this morning from Zubeida Cruz-Monserrat. Just a brief reminder, we have different adipose tissue depots in our body, and I will be focusing on white adipose tissue, the largest portion of which is subcutaneous adipose tissue, but we are all interested from a metabolic standpoint in the visceral adipose tissue. And I would like to remind everybody that the visceral adipose tissue, as we define it, is whatever it is inside the muscle walls of the abdomen. So the retroperitoneal is still visceral adipose tissue, and that is where the pancreas sits. And there's a little bit more literature about the perirenal, very little, I would say, about the volumes of the peripancreatic adipose tissue, because it's not easy to define that. With that in mind, we have the landscape of pancreatic adiposity is somehow different because it includes peripancreatic fat, as I said, and there you see an organs that we receive in the lab, an organ from a human organ donor, and you can see that this is the adipose tissue that is in direct contact with the pancreas. The pancreas, as you know, does not have defined capsule, and sometimes this adipose tissue kind of infiltrates the pancreas. We don't exactly know the mechanisms in humans, the mechanisms of development of the intrapancreatic fat. The intrapancreatic fat is also convoluted, if you wish, because we have the interlobular adipose tissue, but also we have adipocytes in the lobuli, and these do not necessarily touch the islets, which opens a question about how is the communication happening, and maybe Manami Hara has something to say because she studied the vasculature so well. Of course, there are extreme situations like in cystic fibrosis where we have a complete replacement of the exocrine pancreas by adipocytes, and so that is an extreme situation that I'm not going to touch. As well, I'm not going to touch the intracellular fat. We know that there are lipid droplets in acinar and in endocrine cells, not necessarily associated with diabetes, and so I'm not touching that for time reasons. So peripancreatic adipose tissue, it is a portion of visceral adipose tissue. We all know that visceral adipose tissue is a marker of ectopic lipid deposition and is an independent factor for obesity-associated metabolic disorders. So, I mean, we all know and heard talking about the metabolic syndrome. Visceral adiposity is one of the features of the metabolic syndrome. We learned this more than 40 years ago. And I mean, there's plenty of studies showing that visceral adiposity is associated with diabetes. So I can end my talk here, I would say, if we are talking about peripancreatic fat. What is happening in this ectopic fat deposition? And ectopic fat deposition, visceral adipose tissue is the main ectopic deposition, if we wish, but even the intrapancreatic is an ectopic deposition. What happens is that the adipocytes increase in size, they have some local alterations, they start producing pro-inflammatory factors, cytokines, they stimulate local fibrosis, which impacts vascularization and oxygenation, so they suffer even more. And at the same time, they call in inflammatory cells, which, and these cells increase the inflammation locally. So this is what we think is responsible for the systemic inflammation that is associated with the metabolic diseases. We don't know, however, if these mechanisms act uniformly across all depots, and if this happens in the pancreas in the same way, because there's very little cell biology in humans. I have to disclose that I'm only touching human studies, again, because I had to make choices for the 15-minute presentation. But there is a very interesting article that came out last year from the University of Colorado group, Brian Bergman, And he isolated, he received from the HPAP group the peripancreatic adipose tissue, and he studied the secretome of this adipose tissue, showing that it produces several cytokines, and compared that with the subcutaneous adipose tissue of a different cohort, and not the same donors. And one of the cytokines that was produced significantly increased was interferon gamma. This adipose tissue is more lipolytic, as we would expect in the visceral adipose tissue, and has a reduced production of beneficial anti-inflammatory cytokines like adiponectin and adipsin. They also challenged the islets. They challenged the islets with the conditioned media from this adipose tissue for only 2 hours. I personally would have challenged them for longer. So they did not see a difference in glucose-stimulated insulin secretion overall, but they did see a difference in males, in using conditioned media from males. We know that there is an heterogeneity of adipose tissue characteristics by sex, and this might be one of these, showing exactly that. They did see some changes in gene expression, and in particular, they saw a reduction of some of the genes that are active in differentiation and proliferation of the beta cells, for example, the MAFA gene. So in my lab, Adeline Divou looked at the peripancreatic adipose tissue of organ donors that we received from the Organ Procurement Organization. This was part of a larger study, and we are presenting a poster looking at this in the context of pancreatitis. But these are just the controls we had for that study, so the controls and diabetic. And we also saw that this peripancreatic adipose tissue has a phenotype that is similar to the visceral adipose tissue, pro-inflammatory, pro-fibrotic. The epithelial mesenchymal transition pathways are a sign of that. On the other hand, we also know that visceral adipose tissue is positively associated with the intrapancreatic adipose tissue. That has been shown in several studies, not always associated with BMI, but visceral adipose tissue and waist circumference are associated with the content of fat in the pancreas. So the question is, are we looking at the wrong thing? Should we look at the intrapancreatic adipose tissue instead when we do correlative studies in the whole population? So we know that intrapancreatic fat deposition is associated with metabolic dysfunction, even though there is some heterogeneity. So and this intrapancreatic fat deposition also increases prospectively the risk of diabetes. These are data from the UK Biobank, and either if we identify a cutoff of adipose tissue content based on the distribution in the general population, or if we just divide, they divide it in quintiles, it seems that there is a dose effect to diabetes onset. But this might still be mediated by the visceral adiposity. So let's look at the cellular level. Here I'm presenting several studies, including an analysis we did in the Human Pancreas Analysis Program data, and we see that in diabetes there is an increase of the proportional surface occupied by adipocytes. Different studies saw this, except the study at the Vanderbilt University, again, Dr. Wright's data, has been cited multiple times. The difference in their study, and I have an hypothesis that I will present and he will comment probably, is that they matched the controls in people with type 2 diabetes, while the other groups had comparable sex and age distribution, but they were not matched. Also, are we looking at the right thing? So the proportion of interlobular and intralobular adipocytes correlate. Again, in all the studies, they are the same. In different studies, they are increased in diabetes versus not, except the Vanderbilt group again. In the HPAP group, we saw something a little strange, if you wish. So diabetes did not have as much intralobular adipocytes as controls with a lot of intrapancreatic adipose tissue. And we looked at this a little bit more in detail, and actually the answer to what was happening maybe is coming from this recent paper the Newcastle group published, where they saw that there are a group of donors with type 2 diabetes with very high content of fat in the pancreas, and on the other hand, there are donors with no fat but a lot of fibrosis. We actually saw that in the HPAP database, and I blocked my scientists from publishing this because I said, well, let's look at this because it's strange. And actually now I understand better the data that we were seeing. And looking again at the Vanderbilt data, they did have a lot more fibrosis in the type 2 group. So maybe they had selected, for whatever reason, a group that was slightly different than others. So this might call for heterogeneity of type 2 diabetes, which we know exists, based on the pancreas structure. The intrapancreatic adipocytes are also associated with beta cell differentiation. There are 2 different studies where they looked at this, published this year, with an they identified the differentiation of the beta cells in different ways, and they kind of reached the same conclusions. How do the adipocytes impact beta cell function? I don't want to go through this complex slide. There are a lot of things that the adipocytes themselves secrete and might be acting. How they reach the beta cells, it's still an open question. But also, are we looking at the right depots? So in my lab, we developed a method. We want to see if the intrapancreatic adipocytes are the same as the peripancreatic adipocytes, because when we do correlative study doing MRI, we capture the intrapancreatic fat and not the peripancreatic fat. So what should we study from a cell biology point of view? So Adeline and Megan developed this method to isolate intrapancreatic adipose tissue in the same way that is done and to study the intramuscular adipose tissue. They just do a microdissection under the microscope, and they were able to do single nuclei RNA sequencing. This is the first case that we show from a normal donor, and we compared the intrapancreatic versus the peripancreatic, and we saw that the peripancreatic is more visceral in this normal case, showing more myeloid cells and less adipocytes, while the intrapancreatic seems less inflammatory in this person without diabetes. However, when we looked at multiple cases, we realized that actually the intrapancreatic seemed to be more lipolytic, and it is even more in the pancreatitis cases that we had. So more to come on this, and we hope in the end to be able to identify the depot that is closely impacting the beta cells. Last thing is clinically, how is that relevant? What can we do to impact the intrapancreatic adipose tissue? There is evidence that very low-calorie diet reduces intrapancreatic adipose tissue, and that is associated with diabetes remission. And similarly, that has been shown in metabolic surgery cases, and that is a meta-analysis of that. And so what about drugs? There are very little evidences of what might be impacting intrapancreatic adipose tissue, possibly decreasing it. GLP-1 receptor agonists, there are no data using the most recent GLP-1 receptor agonists. There's some data using SGLT2 showing an evidence. Insulin and corticosteroids, for example, increase it. So still more to come to try to understand how we can impact that. So in conclusion, we have a lot to study. The visceral, peripancreatic, and intrapancreatic adipose depots are closely intertwined, and so we need to try to understand who's doing what. And of course, we have data and we have access to organ donors, so if anybody has ideas, we are happy to collaborate. And with that, I thank you for your attention. I thank my founders, and I thank the teams I work with, of course, at Advent Health, Adeline in particular, because she did most of the work, and the colleagues at the University of Florida and at the Cedars-Sinai. So, and come to see our poster tomorrow. Thank you.
Thank you, Dr. Kasu. Our next speaker is Dr. Teresa Mestraki, who is Associate Professor at Indiana University. She works on pancreas development and beta cell regeneration, and she'll be speaking on resources in endocrine and exocrine tissue research in diabetes, NPOD, HPAP, HIRN, and others. Thank you.
Thank you. I shortened the title only because it was very long. All right, so we're going to jump right in. I'm assuming green arrow is advanced. Beautiful. All right, so I think what we've learned already, although there has been a significant amount of focus on diabetes today, is that we have a great breadth of what is considered pancreatic disease research, right? And we know that this involves both exocrine and endocrine. They're highly intertwined. And so a lot of these diseases have components of both endocrine and exocrine disease, right? Even though it might be classified as one, there's obviously going to be something that relates to the other side of the pancreas. Other side, right? All right, so what I've been asked to talk about are the potential tissue resources that exist out in the world, and these might be known to many of you, so I apologize if this is kind of something you already know. But in the event this is the first time you're hearing this, please know that these are resources that anyone can access. So at the end of this, I'll have names of all the people that have helped me put this together, and you can reach out to them directly to be able to gain access to these tissues. Okay, to help you understand a timeline of where we are and how we came to have these tissue resources, the originals, I'm aging myself, but it was when I was in my postdoc, they started the NIH-funded Beta Cell Biology Consortium. It was an NIH-funded group of investigators, all that had UO1 grants, that were studying mostly mouse models of disease, but with a focus on understanding how the pancreas develops. That has turned into and kind of helped to spur the creation of certain tissue resources and other banks of antibodies and tools that help with the study of pancreas disease. So the IIDP was created a little bit after that, and within the next 10 years, so was the Network of Pancreatic Organ Donors with Diabetes. And then the next generation of the BCBC, when it was decided that the NIH wanted to focus more on human disease as opposed to mouse diabetes, that's when the generation of the Human Islet Research Network, or HIRN, was created. And this is carried on now to this day. And most recently, we have CARE T1D that's also being put together to identify and collect donor tissue that relate to larger aspects of disease. Okay, so what I'm going to focus on today are where you can possibly get tissue, so these sources of tissues, some interesting things about what these tissue sources can provide, and then also focus on one of the, I had some check marks, there they are, one of the actual online repositories, and there are many, so I'm going to focus on the Pancreatlas just for a couple slides, because it's one that I personally have used and I really love. But all of these on the right-hand side are tissue resources that have data that's been compiled and that you can look at today. You can go back to your hotel room and start, combing through the data. Okay, so let's start with NPOD. It's the one that I'm personally involved in through one of the working groups. So I probably have the most knowledge of this one, but I'm going to try to do my best to explain all of these tissue resources. So how NPOD works, and sorry that slide got a little bit messed up, is that there's obviously interaction between organ procurement groups and the Network of Pancreatic Organ Donors Consortium. And so the organ donors will be identified either for transplant or for research. If they're deemed appropriate for research and not transplant, then that's when they come to NPOD. These are obviously, there's a focus on type 1 diabetes, but I'll show you that it's not just type 1 diabetes that they have organs for. They have autoantibody positive, first degree relatives, healthy controls. They've got some related diseases like cystic fibrosis and also type 2 diabetes. So once they're identified for research, which is here, they're going to come to NPOD, and they've set up a pipeline already where there's going to be a tissue prioritization committee that determines somebody who applies for a project, if the project they're doing actually is appropriate for the research or for the tissue use, and then what tissue they can use. So if that happens, then all of a sudden you are given tissue from the that's been processed through the organ processing and pathology core, comes to the investigator, and we hope that this pipeline is then what happens. And the last 17 years would suggest that it does. There's been a significant number of publications that have happened that have used these tissue resources. So there's about 700 donors that have so far been collected into the NPOD donor bank. The tissues include pancreas, spleen, lymph node, blood, bone marrow, and thymus. It's considered the world's largest type 1 diabetes research project, and this is what they gave me as a breakdown of what you can see in terms of the distribution of ages for type 1 diabetes, type 2 diabetes, there's some cystic fibrosis, and then you'll also see that there are controls that would be able to be sorted that could be matched to go along with these tissues. Okay, so from that, it's been incredibly productive, right? So all of a sudden, it's taken a long time to get these tissues together, but people that are using them and understanding a lot of really interesting things. 380 NPOD projects have been initiated, and many are completed, and they've, from that, have discovered some things that perhaps now we take as like fundamental understanding of human pancreas pathology, as well as even just human pancreas development. So understanding heterogeneity of disease, how there is beta cell loss that happens with duration, growth and replication potential of beta cells, all of these things have been able to be studied because of the source of the NPOD tissue. They've more recently started creating, and maybe you can't read what's in those boxes, but I direct you to the website down here for the publications as well as more information on the key questions. They've decided that we need to go beyond just let's study the beta cells, let's study the islets or the lack of islets, and so here they're focusing on certain key questions that they think are going to be important in helping us understand the whole disease, right? And Melina and I are co-running the one on the exocrine disease, and so if that's also something you're specifically interested, you can talk to either one of the two of us. To give you an example, and now you've seen this graph 3 times, to give you an example, but Martha Campbell-Thompson was running the pathology core through NPOD, and so a lot of how this started, and this as well as her previous publication, a lot of how this was just by looking at the pancreas that comes into their pathology core. And this is an example that was given to me by Irina, to show you a pancreas with diabetes and a pancreas from a donor that does not have diabetes, and you can obviously appreciate there is a difference. So simple things like that, just direct observation, even weighing the pancreas, then led to these really interesting observations and discoveries that have clearly been taken off on, as you saw from Jordan's talk. Okay, so let's flip to our neighbors to the north. I talked to Dr. Patrick McDonald, and he gave me some information about the Alberta Diabetes Research Institute and their islet core. Similar to NPOD, well, not similar to NPOD, but in general idea is similar to NPOD in that they are isolating islets, banking, and distributing islets for the purpose of research. They actually do it in-house, though, instead of coming from organ procurement centers. And so they have, at the moment, it's a bit of a smaller group, but they do 50 to 60 organs per year, and they range in age from young to old, non-diabetes, type 1 and type 2. And this is just some really cool pictures he gave. This is actually Pat right there. So from when they get the pancreas to the islet isolation, you can see that they've got a really sweet thing that's set up. If you wanted to scan that, it's going to have a lot more information from his group. They also, what they've done is initiated a website where you can go to and actually look at the data sets and the analysis tools. And so this I thought was really neat. Before we get to that, though, I'm just going to give you more, on the numbers. 605 donors, which have so far supported 170 research groups across multiple continents. They ship them worldwide. So again, just not being in Canada doesn't preclude you from getting their resources. They have both fresh tissue as well as bank tissue. Pat says they're willing to work with anybody if they need to do a special sample prep. They're happy to amend to be able to help facilitate the research. But this is what's really neat. One, they're funded completely by cost recovery, so a lot of the banks that we have in the US are grant funded. They do have some grant funding, but really for the most part, since, 2018, they've been cost neutral because they've been able to do that through cost recovery. Okay, so this is the cool part. They've put together this website that allows for data sharing. So everything that they've produced, and this is not a small amount of work. You're looking at, oh, my goodness, sorry. Here we go. Analysis of single patch seq data. You can analyze the data sets they have based on phenotype and other parameters. You can look at relationships between genes and proteins. You can do your own pathway analysis. Each one of these is actually a movie, but just if you go to the website, you'll be able to actually navigate it yourself. If you wanted help with that, Pat would be able to help you with that. You can reach out directly to him. But they want people to use this data. So I think the gestalt of what we figured out from all of these repositories is that we don't want the pancreas to sit in a freezer. We want it to be used, and we don't want the data to just stop at publication. So if somebody can use a piece of data, what I think is being done within the community is it's attempting to be distributed as much as possible. So that leads me to the Integrated Islet Distribution Program. It's based in the US, and its mission is to isolate human islets. They also have linked biospecimens as well as integrated data. So this shows you a really beautiful map of where all the islet isolation centers come from. So far, they have done almost 2,600 islet isolations, right? So 300 million islets that have been shipped, again, worldwide. This is not just based for US investigators. They're supporting a significant number of studies. And so this is their workflow. They get the tissues and their islets are isolated, as well as other tissues. I'm going to talk about that. They're distributed to the world. They have associated UNOS and islet isolation center information that's collected with it that you would get with the islet isolation donor tissue. They have phenotyping that happens at Vanderbilt, and then they have genotyping that happens at Stanford. Both of these two things can also be accessed, and that's through their research data repository. So what you can achieve through submitting for a proposal to receive IADP-related tissues would be either human islets, histological slides, or non-islet biospecimens. And that's actually what I want to talk about just for a minute. I figured given this group, we might not necessarily be so islet focused. And so this is actually an underutilized resource, is what I hear from Joyce. That they have acinar tissue that they have associated with almost every single islet isolation that they do, as well as blood, serum, plasma, spleen, peripancreatic lymph nodes. Like there's a lot of non-pancreatic or non-islet tissue that is up for grabs. And so what she even did was they gave me how much it costs. And what they're saying is that it could be if there's only one person that wants it from one donor, there's the entire volume of tissue there that's open to you. So really, these are not distributed exactly the same way that the islets are distributed. They don't really have a way of addressing what the quality of the tissue might be, especially for the acinar tissue that just doesn't have the same kind of historical measures that islet isolations do, but they obviously have it all, and it would be something that could be able to be used in study. Okay, so last, I've got like a couple more slides. This is to give you an idea of what one of these data repositories would look like. And the one I'm going to show you is the Pancreatlas. The mission there is to build an atlas of human pancreatic images. And so what this group has done, it's been spearheaded by people at Vanderbilt, Marcela Bersova and her team, is they've collected images that have come from a number of different sources. So these are people who have done projects that have generated stacks or 3D projections of tissue, traditional immunofluorescence, whole islet scans, or even multiplexed immunofluorescence. And what they've done is put this together into a website. You can access that data. The Pancreatlas is what this QR code here would get you to their website, and you're able to get any of these 4,300 full resolution images that come from all of these other studies, and you can ask your interesting question, right, and possibly utilize the data in a way that hadn't been thought of yet. So just please know that that's out there. There's a lot of different, there are a lot of different manuscripts that go along with this, and some of the data can obviously be linked to the original phenotyping places that produced the data. One in particular that's really interesting is this Handle P. It is one of the websites that has data that I didn't talk about, but it's up here. And this one's really unique because it's neonatal development in early life. So these are human tissue images that come from pancreas from neonatal donors and donors from early post-birth. Okay. So with that, I obviously cannot take credit for any of this. I am merely the messenger. So if you're interested in anything related to the NPOD, Mark Atkinson or Irina are the two that you can reach out to. They direct and or run this. Pat McDonald is who runs the Islet Corps up in Alberta, Canada. Joyce Nyland would be the contact there for the Islet distribution program. And then this website, Pancreatlas, was the one I focused on, but there are actually, like I said, 3 others, and I'm happy to pass along those other websites if anyone is interested. Okay, that's me. Thanks.
Thank you, Dr. Masra. Please. We'll request that the speakers please come to the podium. The session is open for questions. We request that those who have questions, please keep them brief since we are short on time.
Yeah, Jordan Wright, Vanderbilt. My question is for Brittany. I'm thrilled to see that you did this study in TEDdy. When I first heard of what TEDdy was, my first thought was that the fecal elastase experiment, but I did not want to handle all those fecal samples. So my question though is, so I presented it as though this is insulin deficiency. I think we have not yet answered the question of that data suggests that this is predisposition, that people are starting with a smaller pancreas early on. So I'd like to hear your thoughts on what your thoughts are on it.
Yes. Sorry. Oh, there we go. Got to turn it on. Okay. Yes. So that is a great question that I'm still thinking about. You know, it could still be insulin deficiency. It could be that they start with a baseline insulin deficiency. You know, it's hard to say for sure. If I had to guess what's going on, I do think insulin deficiency is the very much primary driver of all of this, but maybe, you know, there's some sort of baseline defect in these early progressors. Now, these are in a unique population that doesn't necessarily define all of type 1 because it's very early progressors to type 1. So maybe these specific individuals have some sort of baseline difference in either pancreas volume or, you know, the number of islets or something like that, and that's different than these, you know, later progressors. But I also do think maybe there is some component of inflammation, as you know, in cohort B, there was a little bit of a difference in the trajectory, but overall not cross-sectional differences. And if there was a difference, I think we would have seen it because I looked at like 4,500 stool samples. So it was a big cohort. And so I do think, you know, your hypothesis of insulin driving like this, I think is probably the main driver still. And there just maybe some like individual cohorts that have a little bit of other things at play, potentially.
Okay, thank you. Wonders presentation. I'm from China. I'm Yongxing Du from the National Cancer Center of China. So my question is for Anna. I agree with your opinion that the adipose tissue might play a role in the diabetes. I do have some experience that I encountered some patients in the outpatients, their CT or MRI scans indicate the fat infiltration in their pancreas, especially for those patients with new onset diabetes. In this way, I agree with your opinion. So my question is straightforward. How can you define the peri-adipose tissues and intra-pancreas adipose tissues? And how do you isolate these tissues?
Okay. So I'm not sure I understood the question, but are you asking how you find it in your clinic, or my method in the lab?
Your methods, how
Okay.
to define and isolate the adipose tissues.
Okay. In my lab, I have access to organ donors. So with the collaboration with the local organ procurement organization, I get the pancreas with surrounding fat, whatever it comes with. And so we isolate the peripancreatic adipose tissue. We try to make sure that it's close enough to the pancreas, but we want to make sure that we do not collect pancreas tissue. And so that is relatively easy. Then we snap freeze, or we do explant culture, so we collect the supernatant of the adipose tissue. The intrapancreatic adipose tissue is a little bit more challenging. We use the method that is, as I said, is used to isolate the intramuscular adipose tissue. And it's done under a microscope. They microdissect the tissue, and they isolate only the clusters of adipocytes. So I'm only isolating the interlobular adipocytes. I cannot isolate or have much information about the intralobular adipocytes. So we tested that. We did single nuclei RNA sequencing after snap freezing the tissue, and we saw that tissue, only 6% of cells were not adipose cells, so they were ductal cells, which is kind of expected in the septi. I don't know if I answered your question.
Okay. Is there any distance definition for the peri-adipose tissue?
No. I would say we try to take 1 millimeter from what we think is the border of the pancreas. Sometimes, particularly in the very obese people, it's very difficult to identify the border of the pancreas. My experience in islet isolation, we never used donors with BMIs above 35. I've isolated islets from donors of 56 BMI, and defining the border of the pancreas in those organs is a little bit difficult.
Okay, thank you.
Thank you very much, panel, for those really interesting discussions. I was going to ask a general question, if I may, and I don't think it's been considered today, and that relates to the long-term consequences of really effective versus not effective blood glucose control. So one just wonders, obviously the exogenously administered insulin is not in the pancreas, but presumably there must be a number of different impacts of tighter control on all of the complications we've been talking about today. I just wanted to get the panel's opinions on that. Thanks.
Yeah, I'd have to go back and kind of parse through the data a bit more, but I think there's been efforts to look at glucose control and its impact on exocrine volume and function. And from my general gestalt, it seems like there's not a lot of impact there, but there are one-off kind of studies that have found impact. So I think that the results are a bit mixed in terms of just simply looking at exocrine function and volume. But yeah, it's a great question, especially with vascular changes for sure. As we heard today, that's super important. And so I do think it's a question that could be explored more.
Could I ask one question from a non-diabetologist? And this will probably sound stupid, but when you have patients, let's say, that are destined to get type 1, and you're looking at pancreas size or acinar cell mass or some other factor, and there's this, I guess what I've heard today is that insulin is an important trophic hormone, and it's important that it's delivered in the gland itself. And but Anna said that when she looked at donors with type 1 diabetes, it wasn't like the acinar cells just got lonely from no insulin stimulus and kind of withered away. They were all blown up. There was fibrosis, there was inflammation. So how did they die?
I think it's the million-dollar question, right? For my friends in type 1. I don't know I have the answer. We know, for example, that cytokines the beta cells are not very happy with cytokines. If you bathe them in an inflammatory milieu, they go in apoptosis, but I don't know if I
think that's an open question.
So, looks like some people are taking off, but I'll be really brief. So I know it's been a long, long day. This has been an incredible session. We've covered everything from exocrine pancreas to type 1, 2, 3, 4 types of diabetes. I don't know if there are 4 types. There must be more than that. So first of all, just some thank yous. First, Melina, as the president of APA, thank you for conceptualizing and organizing this session. I think it was incredible. I might think of this area as an emerging topic in medicine that's poorly understood and may have a big impact. Maybe we can answer what Chris's question was. Why did I'm not sure if it was the exocrine pancreas dying or the people. I'm not sure what you were getting at. But I think there were some just amazing observations that I took a whole bunch of notes and I'm trying to organize it in my head right now. But there were so many amazing observations that can lead to better understanding. Just a couple that come to me. I mean, why with type 1 diabetes do you get fibrosis and exocrine gland loss? I mean, it's not just a lack of insulin. It seems more complicated, for example. Or, why do you get diabetes with pancreatic cancer, which I was interested in, and the studies showing you get inhibition of insulin production and glucagon changes. So why does that happen? What are the etiologies of that? So those are just some questions that came up. I was really fascinated, too, by the first talk by Katie, where there was what she talked about is a demonstration that ductal cells can regulate the beta cell, and that's truly, out of our minds. We don't think about that. But there must be mechanisms that explain these things. We just don't understand them very well. And I think from my point of view, it's just about learning and trying to figure this out. And hopefully this is a session and we go back and we start interacting with each other. I think it was pointed out several times during this conference that it's a team sport, that we require all kinds of disciplines in order to understand these areas. And it's extremely important. So with that, also in addition to thanking Melina, I want to thank, all the speakers and the moderators, that truly great discussions, and the audience for participating and asking questions. So again, thank you, and Godspeed, and let's start interacting with each other and figuring these problems out. So thank you. Bye.